me, babe. I'm falling. Come and save me, baby. I'm calling. Come and catch me, babe. I'm falling. Come and save me, baby. I'm calling.
Coming to this session. So we have a very interesting session, and this is the first time we are having this session in our Research Summit, Biotechnology. So we have, as you can see in the original program, we had 5 presentations, but unfortunately Orkan Sawyer from University of Warwick has emailed me, and he had a family bereavement situation, so he had to cancel his trip to here. So we have 4 interesting presentations today. The first one is from Alistair Russell from Cancer Research UK, and I think you will enjoy it. So he's going to talk about CRISPR.
Brilliant. Thank you, Emre. Thanks very much for having me here. This is a little bit outside my comfort zone, and hopefully not too far outside your comfort zone. So let me just make sure that I can see if that works. Yeah, is there a laser pointer?
I haven't tested this one.
That's okay.
Yeah,
that's all right. This one? Yeah, that works perfectly. Thank you. So as Emre said, so I come from Cancer Research UK, so I'm a little bit out of this sphere. But today I'm going to talk about CRISPR. That's what my lab really specialises in. And the reason I'm here today is because this is a technology that's really breaking down traditional barriers between disciplines. So really, this is all I'm going to talk about today, this slide. The molecule of DNA. It's the one thing that unites every living thing that we know about in the universe. Every living creature or organism, what have you, in the knowable universe is united by DNA as a storage system. And on that DNA, it encodes all of the different attributes that make us human, that make me, that make you, and make us different from each other. Coding all sorts of interesting attributes such as height through to intellect and beyond. So the story started long before this, but in 1953, for our purposes, it really started here with Watson and Crick when they uncovered the structure of DNA And that really just gave us some sort of insight into DNA and really led us to understand that DNA was a storage medium that connects all living things on the planet and gave us insights into how it's copied. And DNA is often called the code of life. Every cell in your body contains DNA, or almost every cell. And that DNA is an identical copy from one cell to the other. In some sort of way, we're all just little bags of cells, all full of DNA. There's 37 trillion cells in the average human body, and about 3 billion letters in every cell of DNA. If you're to add those up, and if we're to look in more detail at these numbers, the 37 trillion cells in the body, I mean, that number doesn't really mean much to anybody. But if I was to tell you that's 800 billion more than the number of galaxies in the observable universe, then you get a sort of a bit of a handle on the complexity of the human being. And if I was to tell you that there are 3 billion letters within each cell, if you were to pull them out and tease them apart, they'd be about 2 meters in length. And so if you were to take the DNA from an entire individual and pull that out, it would go from here to the center of town, out past Pluto. So that's just one individual. And so that's the amount of information you have stored in your body right now. But of course it's not stored. It's not a static thing. It's not a static chemical. It's constantly remolding, rebuilding, what have you, copying itself. And so every time one of your cells dies and gets replaced, every time you start to grow or what have you, or shed off some more skin, that DNA is being copied. So to put that into some sort of context, in the last minutes every single one of you has made 300 million new red blood cells. That's shooting around your body, carrying oxygen from your lungs to your tissues, hopefully to your brain. There's 12 million new gut cells digesting your lunch, and 40,000 new skin cells adding to the dust in the room. So all of that together makes 1 exabit worth of code that you've all just produced unknowingly in the last minute. So that's just to really put that into context that DNA, it can be vast, it can be complex, and it can be rapidly remolding. So I'm just going to take a step back and look at the field of molecular biology and how we've interacted with DNA over the recent and more distant past, and then I'll come round to CRISPR after that. So DNA, really the focus on DNA over the last 50 years has been primarily on being able to read it. And so we read it through sequencing of DNA. I'll talk to you about that in a moment. And so what this little pictograph shows you is sort of the history of us being able to grapple with and read DNA. So back in 1953, our story really begins, like I said before, with Watson and Crick. And through a series of Nobel-worthy discoveries, right up until about 1986, we developed the technology to be able to read DNA. But we still hadn't read a human's DNA. And so that began the big endeavor of the Human Genome Project, which lasted 15 years. And that was really a flat-footed race between a private firm and public, and academia. At the end of that, we ended up with our first complete picture of what the DNA in a human looks like. And then just 3 years after that, all the technology that was used to create and derive that human genome was superseded by next-generation sequencing. And I'm just going to put that into some sort of context here. The Human Genome Project was a massive global effort involving hundreds and hundreds of laboratories throughout the world. It cost $2.7 billion on average, as an estimate, and took 15 years. Now, if we're to look today at the latest sequences, this one here, for example, which we have about 6 of in our institute, to sequence a human genome, it doesn't take one machine, it takes one small lane in the machine. It costs $1,000 and it takes about 72 hours. It actually can take a lot less than that. So really what I want you to appreciate from this isn't all news, this is just to allow you to appreciate how the ability to read DNA has gone from something sort of archaic in an exponential fashion to something that is fast and cheap and affordable and scalable. writing DNA is something that is a field that's sort of come along a little bit slower. And so to be able to write DNA, what that really means is to take a bunch of chemicals, put them together in a test tube and construct DNA one base at a time. And so this only just really started setting off in the 80s, so 30 years after trying to read DNA. But within the last probably 3 to 5 years, a series of startups and industry sort of focus has occurred in this area specifically for reasons which I'll go into very soon. And what we're seeing now is a massive increase in the rate at which DNA can be synthesized and a concomitant reduction in the cost of synthesizing DNA. So one of the main drivers for this is the obvious utility of DNA as just a data storage medium. And the reason for this is the highest density, large-scale data storage medium that humans know about. You can theoretically put 215 petabytes of information into just 1 gram. So it's also the most stable data storage medium known to mankind. At the moment, this is our most robust and reliable storage medium. It's magnetic tapes, and we rewrite those every 15 to 20 years. But if you go into the Houses of Parliament and look what they store their information on, they store them on parchment because that lasts a lot longer. That lasts for hundreds of years. So this is a picture of the Magna Carta. However, DNA, we're extracting DNA from ancient fossils, from the permafrost, from woolly mammoths, and so what we've got here is we've got something that can withstand thousands or tens or hundreds of thousands of years. So we've got this high-density data storage platform, which is incredibly robust and incredibly versatile. So already scientists have encoded the whole of War and Peace into DNA, the entire contents of the seed vault, and Microsoft just recently encoded OK Go's music video into DNA. So, I mean, there's definitely a keen interest from companies such as Microsoft to at least store some of their cloud storage information into DNA, and that's a proactive area they're looking at. But from our purposes, this is all sort of old news. This is in vitro. We call this in vitro. This is non-living systems. So what I'm going to really talk to you today about is taking all of these advances and using the technology known as CRISPR. We're starting to do these in vivo, so in living creatures, living organisms. So it's a massive paradigm shift. So what I'm going to talk to you about briefly today is the genome editing revolution, explain a little bit about what CRISPR is and how it works, and then talk to what the future holds. So this is a graph of just Google search terms of CRISPR over the last decade, and all I want you to appreciate for this is that it's increasing. Some event happened at 2013, which we'll go into in the next slide, and that set this skyrocketing. You could see that at 2013, CRISPR was really the domain of Cambridge here and Cambridge over the pond. But by 2017, the distribution has really gone throughout the Western world. Although New Zealand seems to be missed out, so I don't know what's going on there. And if we're to give some sort of comparison by some sort of search term that we're all familiar with, you can see that CRISPR is eclipsing even the zombie apocalypse. So I mentioned 2013. So in 2013, I don't want you to really read and understand these scientific articles, but 3 publications came out that just really changed the way and started a field, and that was these 3 here. And all they did is they took a bacterial immune system and they used it as like a rewriting tool to rewrite the DNA in a mouse and also in human cells. Just 4 years later, which in biological research is a heartbeat, all of a sudden human embryos, fish, crops, woolly mammoths, etc., have all had their DNA rewritten. I've highlighted some of these things, human embryos, woolly mammoths, and dinosaurs. I'll come back and talk about human embryos later on. But this is the first time we've been able to design or develop or maybe exploit a technology that's completely agnostic to species, that can cross all the species borders. So people are using it, like I said, in agriculture to rewrite the DNA of animals or crops and what have you. But also we can look at it in human embryo editing for maybe the eradication of disease or creation of designer babies. There's a lab in the US who has coined the term de-extinction, and they're trying to bring back the woolly mammoth. So they're fusing woolly mammoth DNA from the permafrost with elephant DNA. And also dinosaur I've got there. So in some sort of scene reminiscent of Jurassic Park, people are fusing, well, predicted dinosaur DNA with chicken DNA to try and recapitulate Jurassic Park. So I think a good barometer of a disruptive technology is how it hits the headlines. And generally in science, this is a long, slow lag between basic discovery and some sort of, well, at least in my field, clinical impact. Usually it's a lag of about a couple of decades. But what we've got here is a lag of 2 years. And this has never before really been seen, certainly in my time. And so here's an impact, a directly relevant impact, which I'll talk about on the next slide. There's talk of CRISPR being heralding a new wave of treatments in the clinic. A couple of studies came out last year, and these are interesting. These are done in animals, animal models of muscular dystrophy. One of the things I said about CRISPR is it's agnostic to whether you're a mouse or a man. So in many respects, any therapeutic that you make towards a mouse will work in a man. And so what they've done here, they've done 2 studies. One where they've taken a mouse with muscular dystrophy, injected the editing reagents into the muscles and cured that. That's in the body of the mouse. That's not in the germ cells that will be passed on. But another group edited the germ cells. So what they've done there is eradicated it from the baby mice that would be born and every mouse for the daughter, the granddaughters, etc. So they've eradicated disease from that entire line. That has huge obvious ramifications for human disease. And then at the end of 2016, CRISPR gene editing was tested in people. Since then, there's at least 25 clinical trials going on with CRISPR editing in people. This is an unprecedented pace we've never seen before. And I mentioned human embryos being edited. And so these have also, over the last, well, twice this year and once in 2015, been edited. So this is something really what I want to get across here is it's gone from a basic science discovery straight to something that affects everybody in this room within the period of just a couple of years. Just to look a little bit closer at this case here. So this is young Layla Richards, and she presented at Great Ormond Street Hospital down in London. She was diagnosed with a blood cancer at 3 months of age. And so she was given the standard point of care, which is a chemotherapy and a bone marrow transplant. However, that failed. After that, there are no therapeutic options. So before her first birthday, so at 11 months of age, she was sent home, and her parents told her to make her comfortable. She was not going to live to see her first birthday. So under some compassionate grounds, and through some sort of serendipity, some research which just assisted technology to CRISPR, was used to treat her. And so what they did here was they took just some healthy immune cells from an American donor, just an American male donor, and these are T cells. So their job is to go out and seek and destroy viruses in they're part of our immune system that goes out and seeks and destroys viruses. So 3 changes were made with the CRISPR-like technology. The first was so that instead of going out and seeking and destroying virus, they would seek out and destroy her cancer cells. The second was to make them immune to the therapy that they would also treat her with at the same time, so that you're not killing the cure. And the third was to make them self-limiting, so that once they've gone and done their business, there's no trace of the cure. So 1.5 years later, there's no detectable leukemia in this girl. So this is someone on a death sentence that hasn't gone into remission. They haven't got slightly less cancer or what have you. They've got no detectable cancer. So this would classify as a cure, and that's something we don't often see in medicine. So clearly that's why Cancer Research UK is interested in this, and as well as many other healthcare funders. So what is CRISPR? It's a battle as old as time right under our noses, literally. It's a battle between viruses and bacteria. So much like we catch cold through viruses, bacteria catch cold through viruses. They're infected by viruses all the time. Here's a picture of a brown bacterium, and it's being riddled with viruses. And the viruses are parasites. What they do is they bind to the surface, then they squirt in their own DNA. I hope you can maybe pick that out there as that bluey-green strand into the bacteria. Why do they do this? Well, I've got this very complex cartoon here. It took me days to do. So if you can imagine, this is a bacteria, and this is its bacterial DNA, and the virus is binding to it and squirting in its own DNA, which is conveniently a different color. What the virus is doing is it's using the bacteria as a machinery tool to generate more copies of its DNA and more copies of its coat to produce more of itself. It then pops the cell, and away it goes. Now, it's a battle. Occasionally, the cell wins. And what it does is it records some of the information from that virus. It tucks it away neatly into a certain part of its genome for later. That's what CRISPR stands for. I'm not going to read it out. It's sort of irrelevant to what CRISPR is really used for now. But over time, what happens after more and more challenges with more and more viruses is you end up with an array of this information that's being coded. And so if you become challenged with the same virus again in the future, with the same color DNA, you have a two-part system which will search and destroy. It will search for a perfect match, it will cut it and destroy it. So that limits the infection. You could think of the array down here as some sort of sequence-specific GPS locator, and the other enzymes as a pair of scissors. What's really neat about this is as soon as that bacteria copies itself and divides and forms daughters, it passes on all of this immunity to them. So it's really an arms race between the viruses and the bacteria. So I'm not expecting you to understand this, but all I want to show you is a more scientific view of it, and less Pac-Man, a bit more science. And so what we have here, this is the DNA across here. This is essentially the sat-nav component, the variable bit, which is completely programmable to any part of the genome. And this big blob around here is the enzyme, which is the scissors. What's unique about this technology is this is cheap and easy to make, and so our sat-nav is very easy to code and recode. This, all in all, makes the system precise, fast, cheap, and allows us to do lots of rewriting simultaneously, and is incredibly customizable. So how are we exploiting CRISPR at the moment? In healthcare, just like I've mentioned with Layla Richards. In agriculture. This is a picture of a double-muscled cow. It doesn't look pretty. If you're the farmer, you get twice as much money for your meat. But this is a naturally occurring mutation that just has happened just through chance in one particular gene. One particular series of letters, really. So scientists can, with CRISPR, you can recapitulate this at will in any organism you want. So what you're recreating here is a naturally occurring mutation at will. So it's not like a GM crop or a GM animal. You're actually just recreating nature, but with more precision. Living data storage, which I'll get to in the last two slides. Synthetic biology, which I think you're going to hear more about from Jim, and augmented humanity. What I focus on in my job is taking diseased DNA and making it normal. Augmented humanity is taking normal DNA and making it superhuman. So I just want to explain this little experiment that was done in Harvard just within the last 6 months. What this is encoding for the first time information into a living biological system and then reading that out afterwards. So if I talk you through it, they started with a picture of a hand, then used some encoding software to convert that into DNA. The DNA was then synthesized and then put into a bacteria. And it was put in an array-like form, just like I showed you in the previous slides. Then those bacteria, anything can happen to them. You can chuck them in the freezer and keep them for later. You can allow them to grow, expand at will. And when you're ready, you can recall that information just by using that DNA reading, that sequencing technology, which is fast and cheap that we already do, and see what you get at the end with a decoding the reverse algorithm to the start. And so this is the first experiment they did. A picture of a hand. This is the original image. Excuse the resolution. And here is a reconstituted image after the DNA had been growing in bacteria for weeks. They weren't terribly happy with this. They didn't think it was that ambitious. They didn't want to just stop there. And so they decided to see if they could encode a little GIF, a little movie. One of the first stop-motion animations from 1870, just 5 simple frames. And this is how they got on. I mean, it's not bad for a first go. There's about 90% of the information is captured. That's pretty good. That's pretty good, I think, from a first go. This is a world first, encoding this into living, actual cells. But there's a very small step from here to encoding every photo, movie, CD collection, etc., and then embedding it not just in E. coli, but in any cell in your skin. So this is some sort of deterministic approach where you decide what you want to put, what information you want to put into a bacteria. What if you could have some sort of autonomous recording going on where the cell or the bacteria or what have you is capturing information or predetermined information and storing that to be read out in a similar way? So this work has already been started, and there's a large push in the field towards that. And here's just an example of it. It's a really burgeoning and developing area, but it's not entirely clear what this could lead to, but this could lead to living sensors that could record what's happening inside a cell or an organism or within an environment. You could think of it as the Internet of Things version 2, a biological version. But this, I mean, I really want to put this up here because last time I gave a similar talk, this was a pie-in-the-sky idea, but now this is a reality, and such is the speed at which the field is moving. So where's the future taking us? Hopefully within 6 months I won't be talking about all of these things, so we'll see. But realistically, I mean, the eradication of disease is being touted as one of the things that CRISPR might bring. Same with augmented humanity, designer babies, novel threats. I mean, we're starting to discuss and tackle these issues because designer babies, for example, is a very real possibility given that we're editing the germline now. Novel threats, that has not been addressed at all. Augmented humanity, there is a strong interest in this from governments and from scientific research institutes. And eradication of disease, this is obviously where the bulk of the emphasis is at the moment. And that's the end, and that's me. Thank you.
Thank you. Let's get the microphone turned on. Questions?
I can hear it.
Okay, great. Could you speak a bit about the
Affiliation, please. Name and affiliation.
Stefan from ARM here in Cambridge? Could you speculate a bit about the limitation? Because obviously the cell will have some protection mechanisms, and you can't just overload the DNA with new sequences you put in because of how DNA folds during certain processes and so on. So, is there any limitations, or can you just do whatever you like to do?
Yeah, so I mean, so that's really why people have started in bacteria, because we don't know what the consequences are, and because you can put exogenous DNA in there, you can put in whatever you want. Coding information, so on the one hand, if you think about the human genome, greater than 90% of it is what used to be termed a gene desert. There's no real known function for that, so you could argue that that's a perfect area to be able to stick a bit of information in, to code information in, keeping in mind that it's high density information, so you can encode a lot in a small space. The counter argument to that is we don't really understand what that area of the genome does, and so I think this is one of the challenges that we'll have going forward. I think that's why bacteria, yeast, etc. are an attractive option for these sorts of things, for sensing environments or what have you. I think realistically what you would probably create is you wouldn't put it into human cells, you'd put it into a synthetic cell that you would create purpose-built for something like that. I think that's what you would do.
Hello, my name is Omar Costilla from the University of Manchester. Just a question about how much of a threat is this technology? Can somebody edit a genome to spread a disease, or what's the limit of or understanding of what this can do, and if governments are doing anything to legislate or do something about it?
Yeah, I mean, the technology is working. It's going, obviously, it's evolving very fast. It's being applied in different areas, and like, for example, people are using these as gene drives in mosquitoes to spread through the whole endemic population of mosquitoes, so sort of breed in different sort of mosquitoes that don't support malaria, for example, to eradicate malaria. But that has knock-on consequences. That's not gone unnoticed by government and regulatory authorities. There's already a lot of, because we're sort of having a second bite at the apple here, really. Gene therapy came off really about 15, 20 years ago with a big kick, hiss and a roar, and everybody expected so many things from it, and so all of the ethical and regulatory considerations the conversations began then. So as a consequence of that, several different barriers were put in place. So if I decide to go and create a virus with anthrax, or create anthrax right now, as soon as I order that DNA from a company, it goes through essentially a security type process, and they search it for known toxins, known viruses that are hazardous to human health. So there are safeguards already in there. What will CRISPR add on top of what you can already do? Well, that's anyone's guess at the moment, but there are already safeguards in there. And things that block the widespread dissemination of, say, anthrax or other viruses or custom viruses will block CRISPR's widespread dissemination to you. It doesn't solve a delivery problem, getting the editing reagents to a person. So a whole bunch of innovations need to occur. It's not going to be that simple, I don't think.
Okay, thank you.
Yeah.
Another question there.
Greg Parks, University of Southampton. I have a question regarding the specificity of the CRISPR. So obviously it's shown to be actually quite specific from research. But I've seen recent work where actually CRISPR has introduced new mutations at various loci, whilst actually solving the location of interest. Could you speak a bit about that? Because obviously that seems to be taking 2 steps forward and 4 steps backwards, if that's true.
I mean, you could think about it as a version 1.0 of the enzyme and version 1.1 and what have you. So certainly the enzymes we use in the lab, there's 2 aspects. There's one depends on the application. If you're creating an in vivo system, so bacteria or an animal or what have you, if you make an off-target unintended error, it will disappear with time because it will be randomly selected out. So there's one aspect there that it almost doesn't matter in some systems, in some situations. And the other is that, I mean, in our hands, we have to try really hard to create these unintended mutations. It's incredibly accurate, and we have to use the fanciest next-gen sequencing technology and sequence as deep as possible just to see anything off-target. We've only ever seen one off-target information, and that's we've probably looked at trillions of cells to find that one cell. What I would say is there's a version 2 of the enzyme out now, and that's what we use. There's no evidence that version 1 was a problem in our hands for unintended edits, but the specificity of this new one, I mean, I don't see it as a big issue. If you're going to put a human application end to this, you would always sequence the whole of whatever you've created. And so you would just take the version that was clean in a way. But certainly we've never seen an off-target edit with our new version of the enzyme. So, yeah. That's right.
Last question.
Yeah, Stefan Arm again. As this is a natural process, I mean, it has happened during our evolution, like very, very frequently in the past. Is there any estimate of how much of our DNA is actually viral? And is there any other estimate of how much this has become actually functional in terms of we are using then the genes or whatever is encoded in there.
So the CRISPR system isn't in human cells, so it doesn't input the viral DNA into the human cells, only into the bacterial cells.
Yeah.
But a good 20 or 30% of your genome is made of virus,
That's the
retroviruses
question.
and things like that. So there may be an analogous system in human cells that has created that.
Okay,
Okay,
thank you, Alistair.
Thanks a lot.
Come catch me, babe. I'm falling. Come save me, baby. I'm calling.
Great, thanks, Emre, and thank you for organising this workshop. And thanks also to Alistair for really introducing engineering at the DNA level. What I want to do now is to shift the focus slightly to think about cells. And to do this, I want to introduce this time-lapse video. It's from the BBC Natural History Unit. You may recognise it from various sort of David Attenborough films. But you'll see here that you've got this leaf-like structure with this stem growing from the end of the leaf. And as it grows, you can see the small nub of tissue at the bottom here, and that will continue to grow and expand to make what becomes recognizable as a pitcher plant. So as it continues to grow, you've got this coordinate activity of all these cells, millions of cells, all coordinately dividing and expanding and differentiating to make what is effectively a coffee cup structure, in the sense that you've got this lid, if you like, a cap on top of a hollow structure. And this process, as it grows, creates this hollow structure, opens up, and then, of course, it fills with water, and its biological purpose of creating a sac of digestive material that traps insects and feeds phosphorus to the plants becomes clear. But this effectively is a coffee cup. And when you think of a coffee cup, there's something like 200 billion coffee cups made each year on the planet. And as humans make these structures, they do it in a very different way to plants, of course. You have a process whereby you've got the mining of materials, the collection of natural resources, the creation of templates, and the automated assembly of these elements in high throughput. And you're making something which is actually quite similar to the biological equivalent. You have in the coffee cup a curved lip, just as you have in the pitcher plant. In the coffee cup, you have these low-density polyethylene linings to stop leakage. Similarly, you have wax epicuticular material deposited on the inside of the pitcher plant to line the insides to make it waterproof. It's actually a very similar structure, but couldn't be made in more different ways. The design and assembly of the pitcher plant is a collective, a social process where you've got the individual cells that make up that, millions of cells that make up that pitcher plant, start off from a small group, maybe a single cell, which then undergo this sequential process of division to create daughter cells, where the division of cells in this cartoon representation involves duplication of the DNA, physical segregation of the DNA, deposition of all the materials that are required to create two daughter cells. And that process that Alistair also referred to, this proliferation, is something which is self-organizing and pre-programmed. And you see the generation, the splitting, the bifurcation of information systems and the propagation of those physical systems through growth. And as it continues to grow, you've got the reprogramming of those genomes. And again, as Alistair pointed out, each cell has an identical genome, but it's in a different state. So as the cells divide themselves, as they proliferate, they adopt different fates. And we know from many years of developmental biology in plants that the individual cells, say for example in a pitcher plant, undergo interactions with their neighbours to adopt a particular fate. And if this schematic cell, for example, is shown to divide, the two daughter cells are shown here, and these two daughter cells are by definition in different contexts. They're associated with different cells, and you get this bootstrapping effect where you've got differentiation or different states of gene expression emerging by this process. And in fact, the DNA code that encodes that shape is the result of a social process. So if you think of that coffee cup and think of its context, it's more the interaction between the elements in the system that drive the differentiation process. And we're thinking of a Starbucks coffee cup as an example, and I'm stretching the analogy here a bit, but if you think of that, the organizational principles that underpin the creation of a biological version of that is more similar to what happened with the growth of the Starbucks network. The Starbucks collection of shops and the whole industry around Starbucks is based on individual transactions. Individuals making choices, exchanging cash for resources, and the propagation of that social and economic network drives the formation of Starbucks from a single shop in the 70s to this sort of dominating coffee culture across the planet. And in a way, that's what's happening in the biological coffee cup. In the pitcher plant, you've got individual cells proliferating, interacting, differentiating to describe its own program. So it's drawn from within by this highly social process. And of course, it gives rise to all kinds of different variations. So the DNA that it encodes those interactions can encode very different endpoints, very different structures. Here we've got a whole range of things which are closely related but give rise to different outputs. And the process of reprogramming that is now a major challenge for biology. And in order to address that challenge, biologists are increasingly turned to formal engineering. And I'm going to talk about plants as an example, but the same thing really applies to animal systems in different contexts. But of course, in plants, crop plants, for example, we know there's a whole series of issues. We've got very slow generation times, generally speaking, compared to, say, bacteria. They're complex genomes. There's lots of redundancy. They're quite difficult to work with in the laboratory. They're complex, and they're hard to actually visualize directly under what we use in modern biology is quantitative microscopy. So they're difficult to use, but modern plants derive from ancient antecedents. So if the planet was 500 million years older than it is today, and you looked outside, this is what plants would look like. And in fact, if you look at the entrance to the Robinson College, in the cracks in the pavement, particularly against the walls where it's a little bit damp, you see relics of these ancient plants. And I'll show pictures of them in a minute, in fact. So this is the sort of form that plants would have taken about 500 million years ago. Sort of flattened structure, no roots, no shoots, no flowers, no seeds, but they have these sexual reproduction systems, spore-based systems at the top of a flattened structure. So they basically drag themselves out of fresh water onto land, and they are dealing with the challenges of a terrestrial environment, but have a very simple genome structure, very simple architecture. And so we've exploited that in the lab. This is the kind of plant that you'll see in the front of Robinson College. It's a weed, obviously. It's something that we use increasingly as a model plant, as a simple form of something to manipulate for engineering purposes. And I'll talk a little bit more about what all this means. So if you look at the top surface of the plant, it has this very simple reproducing structure. So you've got these little chambers, and these donut-like regions correspond to air chambers. So if you look at that top surface of the plant, each one of those is a modular structure, a bit like the equivalent of a leaf in a lower plant. And so each one of these structures involves this chamber-like architecture. The donut-like structure is this air pore on the top. There's gas exchange and photosynthetic cells in the middle here. And so that's the equivalent of what would be a leaf in a higher plant. You have no roots, but these individual hairs at the bottom part of the plant. You've got the ability to propagate not by making flowers and seeds, but rather by making these features which erupt from the surface of the plant and grow into these sexual features. And they make male and female parts. They're haploid. They cross and create spore capsules. So each one of these little yellow structures here is a package of spores which contain these single cells, which look like this. So there's something like 250,000 of these single cells inside those spore capsules. And when we think of seeds, this is the equivalent in lower plants, where they'll cross and they make millions of progeny. And that's how they manage to get around, of course. They propagate. And each one of these little cells is a package of DNA and resources which allow it to grow. So if you put it onto a sterile media, it grows into a cell, which looks like a recognizable plant cell. We now have green chloroplasts. Within 2 days, it's gone from this, which is one of the ungerminated spores, to this, the first cell division. And that division process keeps going. And just as you watch it, it will divide under the microscope and create this larger scale structure. And after a few days, it ends up as this one of these little structures here. And this is on an agar plate. So it's a bit like those of you might be familiar with yeast or bacteria, growing them on agar plates. This is doing the same thing in a plant context, but making a baby organism rather than a colony. And so it's very useful and accessible for experiments. And the thing that's driving that whole process is the genome. And as Alistair referred to the human genome, here we have the genome of one of these primitive plants. It's much smaller. It's 280 million base pairs instead of 3,000. There's about 20,000 genes, so it's actually not that much simpler, but it has a more redundant genome with genes involved in the cell-to-cell arrangements and how it defines its morphology. So it's a relatively simple and accessible system. And what the field is now doing is, with systems like this, is to adopt more systematic ways of trying to rejig, resynthesize, and reform the whole genome in a way that you can reprogram growth, development, and metabolism. And so we've actually taken some inspiration, or see parallels at least, with what happened in early microelectronics. And this is the first integrated circuit from Jack Kilby and what became Texas Instruments in '58. And so in these early days, these are obviously crude, bespoke devices, but within a few years, by the early 60s, you had planar transistor technology. The first simple integrated circuits were made. You had the adoption of both electrical and physical standards that were used to encapsulate and modularize those components for circuit assembly. You saw the development of both technologies for software design and automated layout of circuits as these circuits became larger in scale. And at the top here, we've got this first planar transistor right through 60s, 70s, 80s, 90s, and 2000s, with the increasing scale of integrated circuits and the number of logic elements involved. And it's on a similar kind of trajectory to the kind of increase in scale that we can work with DNA now. And so modern circuits, of course, have billions of elements in them. And it's beyond the kind of an individual to deal with that. You need these kind of automated skills. And that's essentially what's happening in biology. People are starting to look at how you can use modularity and engineering principles to deal with the kind of scale of complexity. And if you think about biology, you've got quite a different set of issues here. So in the case of a circuit, for example, you've got a layout which is dictated or constrained by insulators and conductors. In the biological context, and this is a diagram of protein interactions inside a yeast cell. So this is the interactome inside a yeast cell with the different proteins. They're all in the same sac. So they all can have the potential, if made, to interact with each other. And what is the determinant here is molecular specificity. So it's not insulation as such, but molecular specificity. And the cellularity provides the insulation. So you've got different layers of complexity that need to be juggled in this case. And so in terms of trying to make the transition between the natural genomes, the evolved information systems which create these organisms, to a more engineering context, this idea of splitting DNA into different hierarchies, so taking natural systems, defining particular functions which can be put together to make logic devices or various types or functional devices, which can then be pieced together in circuits and systems and then ultimately implemented in multicellular systems, is something which is part of the synthetic biology paradigm, if you like, where people are adopting engineering principles or attempting to in a biological context, using DNA as the code effectively to drive these processes. And we have the benefits of abstraction and decoupling of fabrication and design, which come with this approach. So at one level, it's sort of a trivial mechanical aspect, but on the other, it's quite a revolution for a field that's been dominated by analysis of natural systems to move towards a more consciously engineering approach. And so this is an example from the same lower plant, as I've given as an example. So one can take, for example, the genome described by sequencing principles, map some of the functions on that genome, and then create databases which describe the genome not in terms of its physical context, but in terms of its functional components. And use this as a way of creating a resource for automatically extracting parts and being able to collate characterization information. So it's essentially a database not for natural scientists, but a database for engineers. And using this kind of abstraction, you can couple that with, for example, standards for describing these functions, not dissimilar to what happened in the early circuit or integrated circuit design field, where you can start to create design systems, and you can see here these 4 base pair elements which define the ends of, and at the top here there's this coloured schematic of an arbitrary and abstracted gene, where you can define individual pieces of genes, functional pieces, in a way that allows you to start pulling them together and assembling them in a particular way. So you can take these arbitrarily defined functions as DNA pieces and use new highly efficient techniques for putting DNAs together. And this is one particular example. There's a loop-like arrangement for piecing together individual parts to make gene circuits. And that's shown, the extension of this is shown on the next slide, where you can use this loop-like arrangement to start out with individual parts, put them into individual genes, string the genes together to make circuits at different scales by doing this iterative process of assembly. And I won't go into the technical details, but this is another step forward in the kind of things that Alistair was talking about. We're speeding up the work and making it independent of individual decision making. The assembly process is baked into the description of the parts in this way. And so the idea, or the aim at least, is to put together ways of working in biology which provide this sort of, might call it the equivalent of middleware, if you like, where we've got public research in natural systems and applications often driven by the commercial world, but in this case glued together by standardized systems and ways of working and open technologies, where obviously many electronic systems are bound together by open systems which bind together sort of basic research, which I've crudely called academia, which of course is beyond that, and gluing that together to electronics or applications at the other end. And in biotechnology, it seems increasingly important to adapt more accessible open technologies for this, because of the implications for these new technologies for sustainable technologies in the third world, for example. So we've, in Cambridge, have been adopting a consciously open approach. In collaboration with colleagues in Stanford, we've developed this legal framework for sharing these individual elements through open MTAs. And again, I can address that in more detail if people want to know more about it. But what I just want to finish off with is this idea that one can start to use or develop parts-based systems, integrate them back into biological systems in a way that starts to iterate this idea of having a design, build, test cycle in a biological context, where most of the biological systems that we're working with require some kind of software underpinning, that we can use these standardized parts to build DNA circuits of increasing complexity, but in order to test these, they need to be integrated back into a biological system and tested in some way. So there's a lot of focus on how you visualize the role of one set of genes in the context of a whole genome in a large-scale multicellular system. And this all comes down to imaging. And I referred to this briefly before, but it's increasingly important. And that's one of the reasons why these simple plant systems are so important. And in the 90s, there was this discovery that many living systems use fluorescence in a natural world to either to signal or adapt light. And there are proteins which are responsible for these fluorescent signals, which can be used as synthetic genes and used to color or decorate living processes. And here we've got tubes of protein down here, or bacteria which are expressing fluorescent proteins shown in a laboratory context. You can use these proteins, deliver them as DNA pieces into, in this case, plant systems, and use them to paint or decorate living processes in living organisms. And these are shown here, where we've got essentially a paint box of elements. Here, there's 2 sets of targeting systems used to label membranes or the nucleus. And you can see there's different colors in these different arrangements. You can use these systems to decorate. In this case, it's a portion of a plant where we've got the stem portion of a young plant, the young growing point or meristem, and to the first leaf-like structures in the young seedling. And each one of these cells you can see is decorated by this yellow color because we're expressing two different fluorescent proteins, a yellow and a green one. And it allows you to then get ratiometric values for each cell. You can work out what the quantitative expression or activity of a gene is in an individual cell, obviously with a field where you can capture thousands of cells at once. So it opens up the door to new ways of observing and quantifying behavior inside biological systems. And back to our simple plant, one of the advantages of this simple plant called Marcantia is that it makes these spontaneous clonal propagules. So in these cup-like structures, you have a bottom well where there's an embryo-like process that takes place which produces these small propagules. These are such a pernicious weed. If you go to a poorly maintained plant nursery, you'll see these everywhere. Once you start watering, these are called splash cups. So if you're watering the plant or there's rain, these small propagules splash out and propagate themselves. So these are essentially small, nicely defined little structures which can be harvested directly from these plants and then produced in large quantities. And this shows one of these small propagules. You can see there's individual cells here. Each one of these sort of circles here is a single cell. You can label the individual cells inside the plants. You can generate, with different promoters from one of these databases, generate a color-coded model for what's happening here. And these, of course, are alive. So if you run one of these under a microscope, you see the growth of these organisms where you can generate quantitative information directly from the material as it's growing. So you've got a simple architecture, the growth processes are continuing, and you can interrogate it. And so here's another one that's a bit older, the same one a little bit older. So you can see the division and differentiation processes directly. And so to summarize, and representing what's happening in the field, there's this growing attempt to try and capture a more standardized view of biological systems, to essentially fit them into ways of working that are more directly accessible, and also accessible to this design, build, test way of viewing things. So clearly the field is starting to move towards a much more engineering-orientated approach to dealing with biological systems and thinking of it from the point of view of components, systems, the feedback relationships that are involved in maintaining larger scale systems, and trying to address this very difficult challenge of how do you take a code, a DNA code, and program a social system in this kind of context, where we have the benefit of molecular specificity and the ability to make things atomically precise via DNA code, but there's this issue of going through different layers of organization, if you like, to get to the end point, which involves DNA, cells interacting, and physical aspects feeding back on the way DNA is behaving as well. And so this is leading, I think, in the future to this idea that we can democratize some of these challenges, if you like, and this is something that represents, again, work that's going on in Cambridge at the moment, looking at spin-offs from this kind of approach and technology, where, for example, we've got bacterial cell-free extracts, which are now accessible, which don't contain cells anymore. They're essentially cytoplasm that's been extracted from bacteria and can be distributed freeze-dried. So it's the kind of thing that you can use in an electronics lab or a physics lab or an engineering lab. It's not stuck in a biological lab. There are all kinds of challenges that we're developing inside Cambridge, funding opportunities and challenges around instrumentation, trying to bring physical scientists and engineering individuals together with biologists, and looking also at the application of these relatively cheap systems which don't require large resources in Latin America and Africa, and building open curriculum materials for developing tools in that area. So I'll just leave it there, trying to outline the challenges of trying to deal with whole organisms, but also highlighting some of the spin-offs, and give credit to a lot of the people who've been involved with the work. And thank you very much.
Any questions? We have time for a couple of questions.
David Wood, University of Wisconsin. My question is a clarification. So you talked about the differentiation of cells in your pitcher plants. And the part that I missed is if the DNA is effectively encoding the behavior, it's identical in all the cells, and it's encoding essentially the program, the cellular automata. Where's the state? How is that encoded for each cell?
So the expression of the encoded information in the DNA is broadly via RNA transcription. So you're taking the DNA template, making single-stranded RNAs, which then inside each cell is then translated into proteins. So you've got this series of downstream effects. And for each, so you have two cells which might be next door to each other with identical DNA components, DNA contents. They can be transcribed or expressed in very different ways. So if there's, in these simple plants, 20,000 genes, in humans, 35,000 genes, they can be and are expressed in different ways in different cell types. And so not only that, but the protein products, the more functional aspects, can also be regulated in different ways downstream of that. And there's this growing picture or view of feedback between the DNA system and what regulates the expression of the DNA and actually the consequences. So there's feedback from the things that are actually made back on the encoding system. So, and that's called epigenetics. So this idea that you've got both things which might encode or act as a memory of environmental changes or other earlier genetic changes can be encoded by modification of the DNA, both the proteins that wrap it up and the DNA itself. So you've got the characteristic of these living systems is that they're driven by feedback, that nothing happens without feedback on the systems that's driving it. And you have this characteristic of self-organization and auto-regulation, which determines all these things, which makes it quite a challenge sometimes to dissect. And that's why in biology there's been the major challenge, I guess, up until recently, has been trying to decode what's happening in natural systems. But as we go forward, some of these auto-regulatory loops, if you like, are becoming more and more accessible and can be modified. But again, there's this challenge between dealing with the code at one end of the scale, the DNA code, trying to mix and match elements which allow different patterns of expression, which allow feedback from that back onto the, within the same cell and between neighboring cells. So it's essentially the same, made that crude analogy, but in the Starbucks case, the process of building the Starbucks chain, if you like, which is intrinsically a highly social process. It's all of our economics, all of our social interactions are driven by agent-based interactions. And that's essentially what, at different levels, is happening in biology. And the self-organizational characteristics of biology are driven by these intensely social interactions. But the way we need to reprogram them is by a bit of linear code. So it's that gap, if you like, the challenge of trying to bridge the gap between the ability to make precise code and the challenge of creating self-organizing social processes out of that process.
Thank you. Any more questions? So, Jim, I have one question. So in the literature, when a lot of people build these synthetic gene circuits, right? NOR circuits, XNOR. These complicated circuits, right? So what I don't understand is, are they deterministic circuits or they are probabilistic? When you run many times, do you get the same answer, or sometimes it works or sometimes it doesn't work, but maybe majority of the time it gives the desired output?
Well, I think
did I express my question?
Yeah, I think so. So there's different levels of programming, genetic programming, if you like. A lot of the early part of the field in synthetic biology has been about creating simple logic gates, effectively using DNA-based components. And depending on the system, so stochasticity comes into play when you've got low molarities and the frequency of events, because everything's based on molecular contact. So if you've got proteins and DNAs interacting to create some pattern of gene expression, because proteins have to make the RNAs that make the downstream proteins, and they're very dilute, then stochastic aspects will start to dominate. In most systems which either are well mixed and have higher titers of elements, then you can very easily use deterministic models to explain what's going on. and they're very effective in describing the behavior. But for example, Sunny Shi's lab in Harvard have done very interesting in vivo experiments looking at very simple processes, the way repressors and RNA polymerases will interact to create transcription of a single gene. And they've done it at very low titers, where the competition between repressors and transcription activators becomes, there's a form of feedback and form of competition that starts occurring when you've got very low titers of elements in the system. And then you get this, you really need to use stochastic models to properly understand it. And that also, particularly when you've got a lot of biology is based on bifurcations. So you've got low frequency events which might cause a trigger. So you can't avoid stochastic effects at the bottom level of this, where you've got molecular interactions. But when you're making something at, like you would be, for example, downstream in a genetic circuit, where you've got a relatively high titer of elements, then the differential equations are quite happily model those things in a deterministic way. So it's horses for courses.
Great.
Okay, let's thank Jim again.
Come catch me, babe. I'm falling. Come save me, baby. I'm calling.
Thank you very much, Emre. It's a pleasure to come third in this session because so you know now so much about various challenges that we face. So my talk will try to bring you up to speed for making both the culturing and the imaging of cells a bit better. At the risk of stating the obvious, I want to build on a few things that have been said before. So obviously, DNA holds the code for each cell. This is then read, and that's a process that takes about 20 minutes to turn the information in the DNA into proteins. And then the proteins diffuse quite quickly within one cell and interact with each other. So one of those very complicated networks. And it's fantastic, the complexity of the interactions that can happen between those proteins. It's not even one-to-one, it's often complexes of proteins. So even more complexity. And those of you familiar with coding can for sure understand how many agents are at play even within one cell. And then cells talk to each other, exchanging both proteins and other small molecules. And that makes the complexity at the level of groups of cells, i.e. tissues, again at another level. Now the timescales build up on this, and you saw the pitcher plant in the previous talk that develops over several days, its pitcher. And the same happens in, say, in mammals when they go from a small embryo duplicating from one cell to many cells in a whole organism. That also takes many days. So you see, there are processes that take diffusion within one cell can take just a few seconds. That's a very fast process, and also protein interactions happen at that time scale of a second or even less. And then you have processes that take about 20 minutes. That means the change of the number of proteins in a cell through the gene expression. That's over 20 minutes. And then you have processes that take days, and that's about it. There's very few. Well, that if you think of kind of collections, so ecologies in the living world, that can have timescales of months, years, or even longer. I've neglected so far evolution, which is another side with its own timescales, and that's measured in generations. And that's how the genetic code gets changed, and then through interaction you select the organisms that actually succeed. So all of this is complicated, and also stochasticity was mentioned before. It's true that at the molecular level you have a noise which is due to number. You then, through those systems of differential equations that were alluded to, you also have other ways of creating kind of triggering instabilities. So you can have deterministic system which still is chaotic or has other ways to bifurcate and take decisions. Anyway, it's the length scales and time scales that I wanted to bring up because depending on what you're trying to understand in a living system, you'll be working in between two scales. You have one scale where you understand something and you try to control the behavior at the emergent behavior at the higher scale, or you'll be making observation at the higher scale in the attempt to understand something at the lower scale. But in living systems, what I've told you up to now is that there are many, many layers of time scales and also length scales that are at play. That's what makes biology really complicated, but once you start reducing things, then you can bring it to the language of physical science or engineering more and more. And what's been happening in biology in the last few years is that the data has become really nice at many of these levels that I've alluded to, and that's brought in a lot more people who are used to working with physical systems, physicists in particular. Okay, so light is a great tool to investigate cells. Obviously it's good for length scales from very big down to about a quarter of a micron. So you won't see a single protein with light, except if you play tricks like making that protein emit its own light by fusing it with a protein that emits light. In which case you can actually localize even objects that are below the diffraction limit of light. So it's the tool of choice for a lot of lab biology, imaging, and generally the interactions with light. And what we've been learning in the last few years is that it's crucially important to, in many questions, to get down to understanding the differences between individual cells. This is true, for example, if you have a tumor, then the cells within that tumor will not be all the same, because evolution and differentiation of cells is happening very fast in tumor growth. And there's many other examples in which, to understand actually what's going on, you need to have the individuals at the cell level. So in volumes of about 10 microns by 10 microns by 10 microns, you would need to know how many proteins of the relevant proteins, their number, for example, or where they're localized in a cell, or how much gene expression is happening of a particular gene that is affecting the behavior of those cells. So phenotyping is the general word for kind of measuring reliably a certain behavior, so giving it some metric, and one wants to do often single cell phenotyping without sacrificing throughput. It may be that you want to measure all the cells in a community, or it may be that you've been given a collection of cells, say from the blood of a human, and only one in a few will be the cancer cell, and you've got to probe many of them to actually get down to the important one that you want to detect and measure. So there's a challenge of kind of automating things. There's a challenge of also being precise in both in the hardware and in the quantifying. There's challenges of controlling the cell environment that I'll come to briefly in the second part of my talk. Generally, a lot of imaging in biology has rotated around a human being looking down a monocle. So that's from 17th century to 18th century, 19th century. But even until, say, the early years of this century, making a digital video involved an acquisition card and a huge pain, and people by and large make observations by eye and record things in lab books. And that also meant that all the instruments kind of still today in many labs, they look like just small evolutions from this instrument here. They're still designed with the potential for somebody to look down into the sample. And this, of course, is not the way you build a machine if you're just exploiting computers and modern imaging sensors. So, kind of, as you all know better than me, digital imaging really boomed in the last 15 years or so, and there's been just amazing progress of CCD sensors and CMOS sensors. So today, obviously, all the images are grabbed digitally, and so this is our instrument today in the lab to do microscopy on cells, and it's a hybrid. It's a bit like a Tesla car. It still looks like the old car, but it's an electric car, and I'm sure electric cars will have their own features which are optimized around really being electric cars in the not too distant future. So this is a transition instrument. It does use the computer a lot. images are grabbed into a powerful workstation, and about 300 times a second, we can do image processing on the stream of images that come in. We can do edge detection, segment objects, work out what's going on, and feedback into all the gadgets that you see on this optical bench. We can basically feedback to temperature, to the microfluidic control, which is basically pumping liquids into where the cells are. We can change the illumination based on what the cells are doing. This is very important because cells actually suffer very often through the exposure to light, and you want to hit them with your probe lights only at the moments when they're doing the process that is of your interest. So you might want to monitor them with a very low light, and then hit them with all the light which is required for fluorescence imaging only at the right time. So these things can now, well, through the work of the last 10 years, we've basically automated this sort of process, which is, it's already a big step because we almost never peer down these binoculars to see by anything by eye. Everything is happening through the computer and with this potential for feedback, which is basically replacing what the person would normally do by checking behavior of the biological system and doing the right actions at the right time. So I'm happy of this, and it has led to new experiments. There's still all these things in red which are not good. It's because this was the first time that we were building something new. This is actually the result of many years of work, mainly of Yuri Kotar, who's pictured here. It's also led to something which is kind of still unique and customized. It's very difficult to give to anyone else because it's all built around, say, one particular stand. We had to reverse engineer kind of the Nikon protocols to make that work. It's massively expensive because we had to buy kind of commercial bits and then kind of work around to make them work. And all those bits had been originally designed for human work, not to be controlled by the computer necessarily. So in that sense, it allows us to do the science, but it's only a step in the direction that I think things are going. And yet, a lot of what we've built, we think can be miniaturized and taken to a next level. Well, this is just a zoom in of one of the devices in which we grow cells. Again, this is basically called 3 well plates, and here we control the temperature, the gas flowing in, also which is thermally controlled. We heat slightly the top surface so that there's no condensation. And these are all kind of the tricks that one has to build in order to do good microscopy, and to let it go, say, overnight or over a weekend without having to be there to kind of keep the thing going. This is a different instrument, which we also partially automated for growth of red blood cells in connection to malaria parasites invading them. And this has its own challenges because it's an experiment that has to last several days because the malaria parasites grow over a whole cycle of 48 hours. So again, it has to be very stable. Okay, so the stuff that we've learned there in the last 10 years, we now want to kind of port forward and miniaturize and make low cost and spread out and standardize. These are themes that you also heard previous speakers talk about, particularly Jim, and in fact, we've had many conversations with him too. But this particular, so I'm going to show you a couple of ideas and prototypes. This 3D printed microscope is the work of Richard Bowman, who is now academic in Bath. And you may have seen it because it's been publicized and used already by several people. So recently we tried to imagine how a battery of 20 or 100 of these would help us with the malaria work. And we're trying to obtain funding still on this. So loading of samples here would still be manual, but then this painful kind of wait of 1 or 2 days could really be made parallelized into 20 or 100 because these things cost a tiny fraction of what the Nikon stand costs. And the image quality that comes out is very comparable. However, you then have another kind of bottleneck, which is the image processing, which at that point, if you're having 100 streams of data with images taken several times a second, really has to be automated. You can't possibly imagine a human intervening to either make the segmentations, detect contacts, measure sizes and stuff. And the data would be so big that you can't even imagine a human curating errors and checking for errors systematically. So connected to this, there's a challenge of computer vision and image analysis that is scaled up compared to working with a single stream, where you can still expect the human to do some checks. Here, if you're thinking 100 microscopes in parallel, there can be no human checks. There's one vision, so moving towards the lowest possible costs in an effort to parallelize up with the challenges that I described. If we kind of take 2 steps of science fiction ahead, then this is a vision that we shared with Emre. We tried to get funding recently, and I think we're still pursuing to some extent. And here the idea is to scale down. So this object is still has a microscope objective there, and it's about the size of a big Rubik cube if you entered from the top there. This object would be even smaller. So we're thinking here, this is the size of a microscope slide, so about 2 centimeters by 3 or 4. And we try to imagine if we were really starting from scratch. So this still has some legacy in it. So this object here is the microscope objective that you would still see in there. Let's see. Okay, here it's hidden under here, and that in itself has basically design rules that are designed for in the end for the image to be the size of a human eye. So you still find human constraints even in that design. Here we try to think, if we're really starting totally from scratch, we're only gonna kind of base things on the fact that a CMOS sensor is going to pick up things. Then how would we do it? The lens could be much simpler. It could look a lot more like the lens in a smartphone. And we could automate kind of error correction and optical corrections and get very good quality through that. We could integrate. I'm going to talk a bit about microfluidics and show you what it really means. But we can basically integrate channels into this modular layered device. These channels can be opened and closed potentially. The technology exists to do that using soft rubbers and kind of pressing down on the rubber. And there'll be kind of control units that control that. And illumination would be very simple because of the compactness, and we would imagine having online pre-processing, because again, now these would be, you could imagine another scale down in cost, because there's nothing very high quality here, and in fact many of the components will be from mass markets. So these you can imagine having thousands of them for the cost of one imaging device of the ones we have in the lab today. Okay, so this is just this is a vision because we can't yet build this. I'm going to take a step back and show you. So this is I took you along the line of imaging and where we want to go with that. I'm going to take a step back and tell you a little bit about how we actually have to handle cells in order to do many of these experiments, again in a generic fashion. So this is true of basically cell biology work, which means a big fraction of biology. It's also true if you think of personalized medicine. If you have, say, a cancer patient who's receiving chemotherapy, then what one would want to do is take a biopsy or a few cells from the patient and quickly test different therapies, because patients react differently to the same drugs, and even the same patient might react differently as things go, or depending on other kind of conditions that the person has. So it's true even in the clinical context that you would in the end do a lot of phenotyping of cells. It may not always be imaging, but a lot of the assays work through imaging. So this is another challenge. It has a lot of technology in itself. How do you handle things? So here I'm going to show briefly a video. So following a similar kind of, there are people who are taking the existing technologies. It's not working. It's not showing you the YouTube stream. Okay, we'll go back to, wonder why. Okay, the YouTube video I was going to show you is the system they have at Luxembourg University, where they've built, they've robotized a whole warehouse essentially with the robot arms picking up the cell culture dishes from incubators, taking them to a microscope. The microscope works as a microscope, and the whole thing is on kind of an assembly line. And that's okay, but that's again the spirit of trying to automate with the instruments and the standards that have been built for essentially human handling. And that for sure is not the most efficient way of doing things. So one thing we that I again want to kind of show you today, and I think is going to be the way that things are done more and more, is scaling down and going to these kind of microfluidic devices and having channels to move cells about. So I'll show you now a few examples of current work. So with bacteria, and particularly rod-like bacteria. So there's a model organism, E. coli, which we also have in our gut, but also is one of the workhorses as a model system in biology. It's rod-like, has quite well-defined size and shape, and similarly Salmonella, which is an interesting pathogen, and Bacillus and others also can be grown this way. You can basically grow these things in single file, in very thin kind of straw microchannels, which are just 1 micron in width and several microns in length. And then there's no space for the bacteria to overtake each other, but they can push each other out. By out, I mean that there's 2 loading channels above and below here, and the bacteria, as they grow, they kind of push the outer ones into the exhaust. And you can maintain this thing with kind of nutrients, foods going in, almost indefinitely for days. And these things have a generation time of 20 minutes. So you can follow hundreds of generations of this loaded device. And imaging, you can probe phenotypes, so information from each cell over its lifetime. It's not very important what we were trying to do here. Ah, maybe. Yes. Okay. That's the power of HDMI. Let's maybe switch the other one off. But you can't see it. Okay. So suffice it to know that an very important phenotyping thing that you want to do with these bacteria is measure the amount of fluorescence. So here they've been genetically modified so that the protein of interest is coming out together with a protein which is fluorescent. By measuring how much fluorescent protein you have, you're quantifying another protein of interest. Kind of similar channels, but slightly bigger, can be used to hold yeast cells. So yeast is good for making beer and bread. And again, it's a model organism in biology. And here we were testing the effects of geometric confinement, so corners on these yeast cells. And so the good news with biology, I told you a lot about the bad news. So one of the good pieces of news is that it recycles machinery across many organisms. And something that looks very different from a mammalian cell, such as a yeast cell, in fact, reacts similarly to mammalian cells, which are more important for us, for example, to confinement. This is a completely different system. This is an algae, a single-cell algae that we started looking at with Alison Smith, who is a colleague of Jim's in plant sciences. The hope here was, again, to be able to watch single cells. So each of these circles is a cell, and this is a channel. It's a big channel. So these are now 10 microns roughly in size. This is a bigger channel with cages, and flow is coming from left to right. To be able to follow these things over their cell cycle. And the problem here is that the cell cycle of these algae cells is several days. So this has led us to develop a standalone microscope, because otherwise we're sacrificing our high-end microscope forever, focusing on these cells. And we also had to invent this way of having cages, because these cells grow in diameter during their life, and then segment up into daughter cells, which then disperse out. So the idea of channels doesn't work well for this sort of spherical cell. So I showed you macrofluidics to deliver cell and to hold them. So holding over a long time and being able to image the same cell is a big deal in many situations. this slide is showing you how we can, another way in which we can move cells about, apart from flow. You can use a focused laser beam which through the forces that come from diffraction of light exerts enough force to move a cell about in a liquid. This is called laser tweezers. And in this image, I'm showing you how malaria parasite, this small round thing here, is brought up to a red blood cell and then kind of sticks, and in this case actually invades the cell. This is the process that malaria parasites do in the blood of people who've received malaria from a mosquito. This looks a bit like this. So laser tweezers, again, up to now, has been a very kind of labor-intensive manual experiment with people kind of through the computer, but essentially manually steering things and moving things about. But it's another technology which is ripe for kind of feedback and just automation. This is the present, and the future might be tidier. This is the present of how we want to grow layers of cells that are as similar as possible to the endothelium, so to the internal wall of a vasculature. And we're doing this again to then flow blood cells and study how they stick to the cells of the vessel. So here we have a big pump to do a perfusion system, and what we found is that it's very important to grow the cells in the presence of flow, because they basically form the correct tissue with the right alignment only if they're grown for a few days in the presence of a flow that mimics what happens in a body physiologically. So that in itself is then the starting point for many experiments. This is an experiment where we're studying the sticking of red blood cells that are invaded by malaria, sticking to this endothelium that we've grown on the surface of channels. This is one of the biggest projects in my lab, and it relates to cells that we have in the airways. Our airways, as you know, when you have to cough, are making mucus, and this mucus is constantly being pushed up, away from the lungs and into your digestive tract. And this keeps the airways clean, and so you can breathe dust and pathogens, etc. And almost all the time, nothing bad happens to you. It's moved, this mucus, by these thin, microscopic, about 10 micron long and 200 nanometer thin motile filaments that are called motile cilia. And there's a whole carpet of them, and they all beat a bit like a Mexican wave of people at a stadium. They beat in making a wave, and this wave is constantly kind of pushing mucus up. There's, of course, diseases where this doesn't work too well. So these are reconstructed shapes from side-on videos of what this is a healthy cilium, which is it's basically doing a nice broad sweep which pushes mucus, and then imagine a whole carpet of these. The unhealthy one is doing a much reduced stroke, and so that comes from a genetic disease that affects the machinery that bends the cilium. Now there's a big physics question here, which is how can we even measure the synchronization of cilia? So to get a good Mexican wave, you need people at the stadium over a long space to be acting together. Here the individual blobs are cells. So this is about a millimeter, and this is just our cell culture in the lab, where there isn't a big synchronization across. And it's a very fuzzy video, and it's difficult to what you see is flickering, but what we really want to extract from this sort of kind of flicking dynamics is over what length scale are these beatings synchronized. So this is perhaps the only piece of information I'll give you in my talk today. It's a new algorithm that we just came out with to get precisely this scale of synchronization. And I think this will be interesting to any of you doing computer vision, because it's a powerful generic algorithm that doesn't necessarily have to be applied on motile cilia or even to cells. It's much more generic. So what do we do to do that? We definitely don't want to go searching for features. We don't want to segment. and but we still want to retain time information and space information. So a powerful way to go about is to subtract each frame with every other frame. Thus, say if I have 1,000 frames, I've now got a million pairs because every frame minus every other frame. But I can then average all the frame differences that have the same time interval. So I go back to 1,000, a stack of 1,000, because I've got the interval 1, the interval 2, the interval 3, etc. And then you can imagine if I've taken frames that are very close to each other, so an interval of 1, and I've subtracted, then my signal will be very close to 0, because nothing much has moved. But if the time intervals are bigger, more stuff has moved if there's dynamics in the video. So that's true up to a point. If you wait more than a big lag time, stuff has moved in the video, but all the conformations have been explored. And so you get a signal that grows and saturates if you wait long enough in this sort of frame subtraction that I've just described. Now the clever thing is to analyze the subtracted frame in Fourier space, so to look at the Fourier modes, and that means searching for how the signal grows in the subtracted frame for each length scale. The Fourier transform kind of selects out a length scale. And if you do that, you get a time scale for a signal to grow that depends on length scale. And the short length scales grow fast. That means the stuff has moved over a short scale quickly. The Fourier modes that correspond to long space take longer to grow and saturate. Now, from how the time scale depends on the length scale, you get a lot of information from this process. And there's been no thresholding, no segmentation, no nothing. Just the video as it is has been fed into Fourier transforms. You can, for example, distinguish whether motion is just random, or if it's directed motion, or if there are vortices or things that have a scale. You will see that in the trend of the time scale versus the length scale. Yes, well, it's almost finished. For the cilia, which oscillate, we get oscillating signals. And by doing this analysis on tiles of different size, that's how we get the synchronization scale out without any user input. So we get a sigmoid that, as a function of the tile size at which we work, gives us the coherence scale. So my last slide before I finish is to tell you that kind of a lot of this emphasis towards low cost and towards kind of building your own instruments, etc., also requires community building. So I have 4 images here. So the thing on the left is a kind of a do-it-ourselves instrument that's working with a yo-yo board. This is a very nice way to connect a smartphone into the physical world. Came out of Google Labs, but SparkFun sells it. It's very cheap. This is a picture of a workshop where we were showing people how to get started with the Raspberry Pi Arduino combos. It was done through the Sensors CDT, which is a doctoral training program with which you can engage. It's based in chemical engineering in Cambridge. This is an image from a workshop that I've been running the last 5 years. It's run by a UNESCO center, and it tries to push these ideas towards the scientific community in developing countries. So you realize that low cost, it's a big deal to us because we can go high throughput, but it's an even bigger deal to somebody who can't even do an experiment unless it's low cost. And this is an image, so we're now trying to get the university students engaged as early as possible, and we're running events for the first year physicists and in engineering, and they're doing something similar with the first years. So I hope my kind of collection of random thoughts triggered ideas, and thank you very much for your attention.
Thanks. So we have time for a couple of questions.
So it's apparent that imaging is important in biology. Are you aware of any other activities around things like cell surface potential or cell resistance that you would want to augment your automation with?
So a traditional and very important technique in cell biology is patch clamping, where you want to see the voltage difference between inside and outside of cells. That has its own very sophisticated hardware, and I don't know how easy it will be to microfluidize it. but definitely, but it can be done also in other ways through fluorescent dyes that report the same thing. So again, imaging often can do things that were being done in a different way before. Another thing where electrical potentials come in is people use impedance to work out whether cells have come properly into confluence, so if they've formed the proper tissue, and that can be included in these sorts of devices. So these devices, people have built electric fields in them, sometimes for the purpose of steering cells, other times for the purpose of probing them or deforming them.
One more question. Okay, thank you, Pietro.
Thank you.
Come catch me, babe. I'm falling. Come save me, baby. I'm calling.
Okay, hello everyone, and good afternoon. This will be this is supposed to be done by my professor. This is my professor's group. IOE is the Internet of Everything group, and we have an ERC project which we titled Minerva, Communication Theoretical Foundations and of Nervous System Towards Bio-Inspired Nanonetworks and ICT-Inspired Neurotreatment. ICT stands here for Information and Communication Theory. There's this big developing paradigm of IoT, and under IoT there's also this alternative paradigm called IOBNT, which is Internet of Bio-Nanothings, which still falls under IONT, Internet of Nanothings, in which we are actually trying to communicate the data from the nano world up to our up to the internet that we know, the digital place that we enjoy. So we want to connect nanomachines and biological entities, which are also mostly composed of nanomachines, to this wider internet. And the unique challenges for this is we need to understand how these nanomachines are communicating with each other on this nanoscale, and what are the techniques they use for networking, and can we actually try to communicate with these networks. And developing the machinery to do that is a major challenge. And the other challenge, after receiving enough data from these networks, the networks has so much data enclosed into a tiny space. The data that you would need to collect in order to observe any meaningful network would be gigabytes per second, maybe even higher, as was mentioned earlier, that you've got so much DNA data, for example, being stored in so little space and so little mass. So Internet of Nanothings was shown by World Economic Forum as one of the emerging technologies in 2016. And then by IEEE Communication Society, IoT, IoE, and molecular communications have been listed as one of the top trends of 2017 in communication technologies. what we know is, what we have come to understand is that we are actually composed of nanomachines that are sort of seamlessly interacting with each other. And there are so many different ways of doing it, from neurospike channels to hormonal networks which go throughout the whole body, and cardiomyocyte channel, which we understand better, for example. And it actually is logical to think that so many networks have found their solutions after so much evolution. They're already presenting us with the best of solutions in terms of networking nanoscale devices and even producing nanoscale devices. So when you think of an artificial nano network, you would consider these, at least this is the smallest bit of the picture, in which you would have a transmitter unit, a receiver unit, and something that they communicate with, which is usually a molecule or maybe multitude of molecules. And there's a channel in which most of the case, it can be airborne, but it is mostly a fluidic channel in which molecules would diffuse. So we have this knowledge, this accumulated knowledge of traditional communications, and we want to actually sort of compare it with molecular communications and see what can we learn from molecular communications in nature, in our bodies, and what can we actually affect by our theories of traditional communications of electromagnetics. So even though everything is very much different, the carrier is electromagnetic waves versus molecules, speed of propagation is light speed versus very low diffusion speeds, and the environment is usually, this is for traditional communications, electromagnetic waves, they could go even in space, but mostly they are airborne. And in molecular communications, we mostly see aqueous, but it can be airborne in the case of pheromones, for example, animals connecting through pheromones. There are examples of nanomachines being built, like by application of external electromagnetic field. You can create these rotors, and this is a, well, maybe I should do this. So this is a smart drug delivery thing. You can think of this as a cancer cell, and if you detect some proteins, your nanomachines would attach, then would put in some RNA or some proteins or some specific molecule. And sorry, this is from the blood vessel, and then these molecules target the cell and cause it to undergo apoptosis. These nanomachines, out of these nanomachines that we might have like this, these are very simple. We then have nano networks, which is a much wider topic, because then you would have lots of communicating nanomachines to perform a common task. And one of the questions is there enough place down there to communicate and do all these? But it's a famous, actually very famous quote that there's plenty of space down there. There's plenty of room at the bottom. So there is enough room for extracting communication networks. We know that. So we say we believe the answers are already inside us, and we are sort of built by these small networks, which actually use the space on the bottom. And well, in Minerva, we are considering to actually find theoretical foundations for we are interested in the neurospike communication in the nervous system. We just dampen down to a specific topic, specific network, and neural network has been chosen. And so we are interested in theorizing, modeling the nanoscale neurospike communication channels, and then after after modeling all the channels, then make it a multi-terminal neurospike communication channel and nervous nanonetwork investigation. So investigate the networks. And in order to validate our model, we want to build this nervous nanonetwork simulator, which is 4 N's. We call N4Sim. Haven't put in here, but we'll see it somewhere. To have a fast-running simulator which can actually simulate large networks of neurons and maybe astrocytes involved as well, and their synaptic communications, axonal propagations, and everything. And after this would contribute a lot in our understanding of the existing mechanisms inside a nervous nanonetwork, then we would like to actually do 2 things. Design ICT-based nanoscale communication systems which are inspired from biology. And then, after designing these ICT-based theories of these communication systems, try to implement the ICT ideas, theory, to envision ways of neurotreatment. So this is something, there's no one way of doing this. You really need to go through the system and see what might be the cause for one problem and try to simulate it, and then try to apply some communication theoretical analysis on, well, the information is not being propagated at this level because of this kind of things, and how can we resolve this? We can actually we believe that we can find ICT-based solutions to cure diseases, neural diseases. And that's actually been going on. So I won't take long. These are the 4 tasks that we had just written in the previous slide. We are getting outcomes. Some tasks are already finished. We are actually in the middle of making the network simulator, and also in the final stage of, we have this proposal of making an artificial synapse, and our plan is to go through and make an artificial synapse. So this is a nanoscale neurospike communication channel. We like to model our nervous system like that, but it's actually even more complicated than this. But the main idea is there's an input neuron which creates an action potential at some time, and that action potential travels down the axon to all the synapses, synaptic ends. And in the synapse, upon the reception of the electrical signal, pulse, a vesicle is released to the synaptic cleft, which is a very narrow band of 10 nanometers across. And those neurotransmitters that are released from the presynapse go and attach to receptors on the postsynaptic wall, which triggers ion channels to open, and ion flow happens. Positive ions flow into the postsynapse to decrease the voltage across the membrane. And these data, all these ion current data, they all come from these dendrites. And there is some sort of dendritosomatic, it's called, calculation that goes on here. And if these ions come and trigger a specific place around inside where this pulse generator is, it causes another synaptic pulse, and then that propagates like that. And this is the basic block of the network. These guys actually, one neuron would communicate to 10,000s of neurons, and in turn receive information from 10,000 neurons as well, in most of the cases. So it's a very non-trivial, highly nonlinear, and very big network that we're dealing with here. And well, we hope that information is encoded in spike trains. We think that, and we actually have proofs of that, but what the coding scheme and everything is, even that is sort of not known so much. So this is just an enhanced picture of what I had shown. These are the specific models that are used in trying to model the behavior of these channels and networks. This is too mathematical and too detailed, so I will not go too much into this kind of stuff. But this is our theoretical modeling that's been going on. We did the vesicle release model, which occurred in the synapse, and the general system model with the type of receptor dependent some specific waveforms that are being sent as ions flowing through. And then we, of course, after getting all these model, we want to actually provide some estimates on what kind of communication channel we have and what's the capacity of the channel, how much information can be put through it. And so these are part of that study. This is a study done by one of my colleagues in which we actually use earthworms as test subjects. I think they don't require any ethical issue at all. You can just do anything on them. Actually, if you cut them, they multiply, become two of them. Very strange creatures, but these are preferred because they are known to have this very large and very simple nervous system to which you can really attach by very simple electrodes. And just to be able to validate some of our models in this basic experiment, this experiment that was done. And after getting all the model done, it's actually one of the important questions we have is how to put them into a nervous network simulator. The reason is the behavior is highly dependent on the parameters of the single neurons and their connections. And we want to be as realistic as possible, so there's this diffusion process that goes on. So we don't want to go through that diffusion process at each time because there are like millions or billions of synapses even in a small network of small nervous system. And running diffusion simulations through all of them is impossible. So what we're doing is we are, based on our theoretical models, we are trying to build up deterministic models that sort of capture the average behavior of this stochastic in nature process and go for the average behavior of these channels. A lot of things need to be incorporated still. One of the most important things is synaptic plasticity, because that's how these things regulate themselves in order to change behavior. And learning happens through synaptic plasticity. And it is really important to pinpoint how these things, how the synaptic plasticity is actually should be seen in the models, in the equations. There's an effort going through that, but what we are trying to do is just put everything in libraries in terms of input-output relations so that we don't have to simulate every cell again and find a way to create a fast-running but still true to the molecular dynamics network simulator. And this is N4Sim. We are actually in the middle of doing this. So there is some simple models. This is one of from the top paper, trying to do this in a case of a model of diffusion-based model of some synapse, which is assumed to be like a real synapse. And also we have done work on in an the channel of an artificial synapse, the molecular dynamics. And the artificial synapse was actually a synapse that is closed from the walls, thinking that if we have an artificial synapse, if we want to sort of bring it about into a living system, it needs to be biocompatible. And one of the most important things is that maybe the neurotransmitters we'll use in that artificial synapse will not be biocompatible at all. So maybe it is a good idea to even also to not to run out of neurotransmitters, maybe make our synapse close and just leave neurotransmitters and suck them back in to the pool where they were ejected from. And that has already shown one thing very crucial, which is the suction of neurotransmitters from the synapse is actually plays a huge role. If the suction doesn't happen, then the capacity of the channel falls dramatically and you end up having no signal information being transferred across that synapse at all. So that can actually, for example, accumulation of large molecules next to synapses can prevent actually molecular neurotransmitters to diffuse out and just kill this link effectively. So this is actually, this shows us one of the ways to have disease, neural disease, which is just having a bad cytoplasm, not exoplasm, which actually sort of blocks all the movement of the molecules. So what do we want to get out of this? One of the major studies that lots of groups actually are going for is spinal cord injury and the treatment of spinal cord injury, in which you hope to get data from above, from the cerebral side of the injury, and get the data across to the below injury side, to the rest of the body, and just repair the broken line, and hopefully the patient will gain its ability of movement. And right now there are many ways that are being envisioned, like epidural stimulation and reading data directly from the brain. But one of our visions is actually just put up these artificial systems which directly take data from one neuron to the next one. But this is a very futuristic thing. But still, even though very futuristic, that is actually sort of being done. There are ways of doing stuff. So this is a way of towards an artificial synapse. You would want to actually sense data and make it an electronical data out of it. This is work done by our friend. He's doing PhD here right now. And these biofets and silicon nanowire fets that he has built actually have the capability of receiving some molecular information. The thing is, which molecular information, how good is the reception? There are so many problems to be tackled yet, of course. And on top of Minerva, we have also gotten this ERC POC grant, in which we are actually proposing to do high-resolution neural interfaces using graphene. Well, graphene has this many things that we like, many properties. you can actually manipulate atomic scale so that you can really have high spatial resolution on graphene. There are many methods that we are thinking, actually nearly all based on graphene. We hope to create highly localized fluctuations in ion concentrations or maybe neurotransmitter concentrations as well, detect them and also generate them. And that would be the basis of communicating with a neuron, actually. This will, of course, will get us towards Internet of Everything vision, which will make possible the bio-cyber interfaces that have so many applications, actually. So this is a childish picture, but this really is the sort of the kind of idea that we have. You can think of these as these. If you see the red lines, these are these small grids, and think of them sub-micron scale. So sub-micron versus sub-micron, and you really want to collect data from one place of the nervous system, and then put it to a lower side, and then restore the communication in the spinal cord, for example. And one of the actually, this sounds very feasible because of the biocompatibility studies as well. So these graphene-based interfaces do not alter target nerve cells study actually shows that graphene is highly biocompatible, and it actually promotes neurite growth towards itself. So one problem in interfacing is when you put something there, you're not connecting to the neurons themselves. But since neurons are dynamic, you expect the neurons to come to you. That's why you need the neurite sprouting. They will come to you and interface with you, hopefully. So in this proof of concept project, we will chase for this high resolution neural interfaces, which is actually a very hot topic. Within last year, actually within this year, 2017, DARPA has commissioned DARPA has funded 6 projects towards high-resolution neural interfaces and spent about more than $100 million just this year, and this is just DARPA. Elon Musk has created this spin-off company called Neuralink, which is trying to do the same thing. So our competition actually in this proof of concept scenario is massive. But it's fun to have. So what are the broader impacts of Minerva? We are envisioning to produce bio-inspired nanoscale molecular communication systems for the vision of IOBNT. ICT-based nervous disease diagnosis tools, and this will utilize Enforcim. Then ICT-inspired treatment techniques. We have ICT-inspired treatment techniques for the normal electromagnetic networks that we have. Similar stuff can be done in human body as well, because it's a communication network as well. For example, jamming communication of cancer cells to prohibit them from doing metastasis, undergoing metastasis, kind of stuff. And thank you. This is the group. This is our professor and the rest of us. And thank you.
Thanks. Time for one question.
I see one question.
Hello, Vihar from Glasgow University. Very nice talk. I like it. In the end, I just saw the graphene. I have other questions about the equations, but anyway, I'll ask you later. How you're linking the graphene with the antennas? They were carbon nanotubes and copper. How you make the contact between the wires and the graphene?
No, actually, that's one of the biggest problems if you are trying to measure something very high resolution, how to get the data out of there. Carbon nanotubes is one option, but there is not much relevant and scalable work which would allow us grow carbon nanotubes on graphene and get them independently. So that's a very hard one. When making, well, there are many ideas that we are chasing, but one of them, for example, the simplest one, just forces us to stay in two dimensions. Graphene is two-dimensional, but we are not gridding in 2 dimensions, but we are gridding only in 1 dimension. And then we can get them out. Otherwise, what needs to be done is there's this other option of using graphene as a membrane, which actually allows ions in and out if you put nanopores on them. So you would create these nanoporous graphene flakes or sheets, very small size. You put them on a microelectrode array and hope that by giving electric impulse you can actually manipulate the ion concentration in the following fashion. If you have holes, if you can calibrate the holes will be large enough for positive ions to pass in, like calcium and kalium and should call potassium and sodium. But chlor or other negative ions cannot go through. There are studies of this, but that's still not scalable thing, but it's still being produced. And what you can do is you can actually put this as a membrane, and if you apply negative voltage, you will actually collect the positive ions to your chamber, and then by an impulse of positive, you can push them out. And you can use this as an idea of, for example, introducing pulses of positive ions into the medium to communicate with the neuron, for example, or to understand what are the concentrations around this neuron, so that you see if there's a spike or something. Yeah.
Okay. Thank you.
Yeah.
Yeah, let's thank speaker again.
I'm clapping myself.
So that concludes this session and also concludes the summit. I think we will have a closing remarks, Chris? No?
Unless you've got some.
No, I don't. Okay, so thank you again. Hope to see you next year.
Come catch me, babe. I'm falling.